start this introduction with a quote that I like from Francis Crick in his autobiography, What Man Pursued. He said, you know, to have a good idea in biology, what you really need to have are a lot of ideas. And I think Umut is a person with a lot of ideas, which can be good, can be bad, but luckily he has the skill to sort of sort through those ideas and pick out a few gems among them in light of the best available evidence. It wasn't, I guess, always so it's nice to see that people learn things through graduate school. I distinctly remember when I first met Umut about 4 years ago, when he came to my office. I had been here at Stanford for about 2 months, and I had an infant son at home who was keeping me from sleeping, so I hadn't slept in about 2 months. So I was in a somewhat poor state. At that time, Umut, who had, I guess, a bit of a rocky start at Stanford, spent the first couple of years making not too much progress on researching lasers, came to my office looking for a new research direction. So I think it's safe to say that we were both in a little bit of a desperate state at the moment. But luckily, since then, it's all, I would say, more or less come up roses. And as you'll see in his presentation here. In his 4 years in the lab, Umut has really covered a lot of ground, and it's really been a pleasure for me to watch him grow into a scientist. So as I mentioned earlier, Umut is a person with a lot of ideas, and quite a few of which have panned out usefully. So the first half of the talk will be about one of those ideas, and the second half, which is published, the second half will be about sort of a follow-up of that, which we're sort of preparing for publication. But apart from what he's going to talk about today, he also has like an extensive amount of other work, which is not going to be included in this seminar. So in collaboration with Aaron Straight, he's dug through a bunch of this high-C genomic chromatin captured data and dug out some evidence of a 50 kb periodicity, which is suggestive of this sort of fabled 30 nanometer DNA fiber. Additionally, in collaboration with Amy Gladfelter, he's done some convincing cell biological analysis of nuclear division in the context of a multifilamentous fungus, providing evidence for the fact that these sort of nuclei within this multifilamentous cell are actually acting independently and push against each other, and has come up with this sort of cells within cells model, which is derived from microtubules and their associated proteins. So all in all, I think highly productive, highly successful PhD, and I think you guys are in for a treat in hearing his talk today.
So thank you very much, Jan. I might need this. Okay. So before getting into the detail of my research, let me briefly introduce my motivation to study biosciences as a physicist. So when I first came to grad school, I realized that we have a pretty well established framework in many areas of physics. That means you know how to think, and even you can predict the outcome of some hypothetical thought experiments. However, I had no idea how to think in complex systems such as cellular functions. Since the framework is not as well established as in physical sciences. But you know, the parts of the cells like DNA, protein, RNAs have been well characterized, but the signaling and the corresponding emergent phenomena is not well understood. So let me explain this with an example. Imagine that you're an intelligent creature living in a far future and inherited all the technology from this era, and without any explanation of how these machines work. And let's say that you want to understand how Apple II works. Obviously, the first thing that you need to do is to look at inside and look at the elements and characterize it and try to figure out what's the connections, what are the connections, and what's going on here, right? Then you might think that you understand how a computer works. But when you encounter an IBM 82 computer and look at its elements and see that the elements and the wirings are different, then you realize that you indeed cannot claim that you understood how the computer works, but instead, maybe you can say how Apple II wiring is. You understand Apple II wiring is. Although the characterization of the elements is indispensable, understanding the computer, how computer works, requires to identify the common working principles with a holistic view, right? So once you know the working principles, you can even design your own computer. So likewise, cell is a complex system that has many interacting parts, which emerges to operate in a specific way. Therefore, to understand the emergent cellular functions, a context-independent framework should be developed. So, a major challenge for such a science, such a research in biosciences, is to understand the signal transduction of cellular functions. And cells execute complex functions based on environmental and internal situations. And these environmental and internal information is the information flow transfer is carried out by the protein interactions, right? So this flow of information is called signal transduction. After many years of research by many people, the protein interaction map has been has been well characterized in details. So here you'll see the protein interaction map in yeast, right? But however, a specific pathway composed of specific proteins is just one solution to an elemental biological problem. Therefore, we need something like working principles of how a cell function, right? For an example, the principles of cellular transitions. And we see the cellular transitions in almost all organisms in various contexts, such as bacterial sporulation, cancer differentiation, yeast cell decisions, etc. Therefore, it's very valuable to have a context-independent generalized picture. So my graduate work mainly focuses on one of the very important transition in the yeast cell cycle here. So we use budding yeast as a model organism besides making bread, beer, or wine. And if everything is permissive, the default mode for the cell, budding yeast, is to divide. And to divide, cells have to overcome many challenges in a coordinated manner, such as they have to duplicate the DNA and segregate the chromosomes and make some more like cell wall, etc. Therefore, we need something. And also, if you change the environment to a not good for the division, then the cells, some of the cells arrest at some point, and the others complete their cycle, division cycle, and then arrest afterwards. So it's been shown by Lee Hartwell that this point, the arrest point, is a decision point, and it's called start. So the summary of my research is in two parts. In the first part, I found that the cell cycle-related transcription only occurs after the commitment, which works through a well-ordered transcriptional program. Then I showed that this is very important for the cells, and consequently, I analyzed different species to show that they also have the same principle for their cell cycle transition. And in the second part, I explained the mechanism of this temporal regulation by using by the combinatorial use of transcription factors and the nuclear organization. So let's get into the details. At the point of start, in the late G1 phase, cells integrate the internal and external signal and commit to the cell cycle if the conditions meet. And this commitment coincides with the transcription of more than 5% of the genome, which ends up a huge physiological change. Although the input signals are gradient and reversible, the transition is highly robust and irreversible. Therefore, for the sake of robustness and irreversibility, a complex mechanism is required to regulate from G1 to S phase transition. And cells achieve this robust transition by engaging a positive feedback into the system. An upstream G1 cycling clean 3 partially activates two heterodimeric transcription factors called SBF and MBF, which are known to regulate more than 300 the transcription of more than 300 genes. And among their targets, clean 1 and clean 2 are two other G1 cyclins, which then fully activates these transcription factors and drives the transcription of the rest of the regulon. And it's been shown, and once the feedback is engaged, the transition becomes independent of the changes in the upstream. And it's been shown by our lab that the commitment point precisely corresponds to the feedback activation. But the story is not as simple as this. So we have, like in the very same regulon, we have a negative feedback element, which is called NRM1, which is negative repressor MBF, which when it's transcribed, it turns off the MBF-based transcription and shuts off the system. So in this G1 to S transition, we have a positive feedback, a negative feedback, and lots of genes with a form and complicated relation. And cells somehow orchestrate this transition in a very robust manner. Therefore, to see what's the principle behind this solution that cells apply, we need some global scale transcriptional dynamics, right, to monitor it. So what people, actually the people generally uses microarray to monitor the transcripts in a global scale, in a genome-wide scale. And what they do usually is they cluster them to group the genes that are similarly transcribed, right? But if we cluster the genes and to look at them, we see, we miss the fine-grained resolution of the transcriptional dynamics. Second, we are missing the relevance of the positive feedback to the transition. Therefore, we need something we need to analyze these transcriptions individually. And for example, we can locate the activation time for each gene differently. Then we can look at the time result dynamics. To analyze the dynamics of the transcription, we used time course microarray data, which is published by D'Italia et al. So the procedure is simple. The cells are grown and arrested in mitosis. Then they're released from mitosis once all they are in the mitosis. And when you release it, the samples are drawn every 5 minutes for 45 minutes. And the quality of the synchrony can be assessed by the budding index measurement. And the mRNAs from each time points are hybridized onto the asynchronous culture mRNAs. And the data is analyzed by a grad student like me. Of course, not all the genes have to be analyzed. Therefore, we chose the genes that are both cell cycle regulated and induced by the clean 3. And then we analyzed 7 separate biological replicates of the time course microarray experiments by a novel algorithm that detects the activation time of each expression profile. Here you see the 7 replicates, biological replicates for clean 2 gene, which are put on an offset to show that that activation time is like pretty reproducibly detected. So if you look at different genes, so by analyzing the activation time, by analyzing this transcriptional dynamics with activation time instead of clustering, revealed an interesting concept. Even though they're targeted by the same transcription factors, the different genes can have different activation times. So this was interesting. And as you can see from the error bars here, we can locate the activation time by using the 7 different microarray experiments within plus minus 1 minute resolution. So if we plot the distribution of mean activation times, we'll see that clean 1 and clean 2, which are the positive feedback genes, are located in the earliest group. And interesting enough, that the negative feedback element comes later on. So you can also plot the same thing with cumulative distribution, which I like more because, first, you don't have to be in the data. Second, you can directly see the fraction of the genes that are activated before a certain time point. But the bottom line is the CLIN1 and CLIN2, the positive feedback genes, are activated first, then the rest of the regulon comes and the negative feedback comes. So we can summarize this with a schematic like this. First, you activate the cyclins, which are like stepping on the gas pedal while driving, and you progress through the cell cycle. Then at the end, you activate the NRM1 and negative feedback, which is like a stepping on the brake pedal, right? So therefore, early activation of feedback ensures that cells first commit, then take the massive amount of cell cycle transcription. So then we can ask how robust is temporal order, right? To see if the results are dependent on the carbon source or the synchronization method, we've performed some more micro experiments. But now we grew the cells in glucose as well as in galactose, and we arrested them in alpha factor. Also, we used another method to synchronize, and repeated the same steps. But here, all these 3 experiments are done by arresting the cells in G1 phase. Now, if we analyze in a similar manner and find the activation times, we'll see that the carbon source doesn't have a global effect on the activation time. Also, synchronization method does not affect the activation times. So if we take the average of these 3 and locate the positive feedback genes, we still see that the positive feedback genes are coming first, then the rest comes. All right, so far so good. But then if we compare this G1 block release experiments with the previously I mentioned mitotic block release experiment, interestingly, we see no correlation at all. So therefore, there's something intervening the activations of those genes, probably due to the arrest phase. So it's natural to ask which experiment is more relevant to the free-running cell cycle, right? To answer this question, obviously, we use time-lapse fluorescent microscopy for single cell. And we requested 10 strains from 10 GFP protein-fused strains from the UCSF library. And with our 2 CLIN1 and CLIN2, we analyzed 12 different GFP expressions. And by using a nice segmentation program, automated segmentation program, we can record the fluorescent intensity by time. And we aligned all these profiles based on their body emergence time. Therefore, we can find the activation times relative to that. And if we take the average of the activation time for different genes, for each genes, we'll have something like this. So the free-running cell cycle expressions are fairly well correlated with the mitotic block release expressions. However, when we try to plot against the G1 block release data, we see a less correlation. So there are 2 outcomes of these experiments. First, it says the protein induction timing is mostly determined on the transcriptional level. Second, the mitotic block release experiment is more relevant to the free-running cell cycle, unperturbed free-running cell cycle. So, all right. So in the first part, we see that there's a well-ordered transcription. Cells first commit, then takes a massive transcription amount. And now we can ask, what's the importance of having the feedback earlier, right? So to answer this question, actually we can elaborate the question more. Like, what happens if we delay the positive feedback? To answer this question, we constructed a strain where the only positive feedback element, CLIN2, is put under a late activated promoter, NRM1. Then now the analogy becomes stepping on the gas pedal and the brake pedal at the same time, right? Just like, note that this schematic is pretty same with the previous schematic. The only thing that changes is we just like put under a late activated promoter. That's the thing. All the network, all the arrows are same. And what happens is something not good for the cells, where you see that the positive feedback delayed case have many problems, like, for example, this cell is like binucleate and trying to divide. These cells are dead, so it's not good for the cells. So when we look at the size distribution, the positive feedback delayed cells are as bad as having no positive feedback at all case. So, but it's a little bit better in terms of viability that having positive feedback versus not having positive feedback. So it's of course, this results suggest that there's a strong pressure to get the positive feedback earlier, right? So we've seen that there's a well-ordered transcription, and this early activation of the feedback is very important. So then we can ask, is it conserved across the eukaryotes? So to answer this question first, we looked at the closely related yeast, Saccharomyces bianus, which has about 65% nucleotide conservation, or like homology. And we've analyzed the microarray data for the Saccharomyces bianus in a similar way. Then we find out the activation times. And when we plot the activation times for the bianus versus Cerevisiae, we'll see a less correlation. However, the bottom line here is the positive feedback elements are still coming in the first place. Therefore, bianus also uses the same logic that they first commit, then they take the transcribe the rest of them. No, the genes are like they're homolog genes. They have very conserved. These are conserved genes. Oh, do you mean this? Yeah, they're actually activated before the first time point. So I'm just like putting it there. Yeah, yeah. That's actually they're activated before the first time point. So we've seen that the feedback first motif is important for yeasts, right? How about the mammalian cells? If you look at the mammalian decision point, it's interestingly they're coming from the different origins, the proteins, but they have a very similar schematic, which is like there's an upstream cyclin D, which phosphorylates RB proteins and relieves E2F transcription factor, which are responsible for transcribing a lot of like thousands of genes. And among their targets, E2F itself and cyclins E1 and E2 and skip2 are the elements that positively feeds back on this transition, either directly or indirectly. Therefore, we have 4 different positive feedback elements here. So then we analyzed 4 different microarray experiments, time course microarray experiments, in a very same way. Then we locate the activation times. For example, here you see the profiles of cyclin E1 with an offset. And if we look at the different genes, we still see the same thing. They're targeted by the E2F transcription factors, but they have different activation times. So if we plot the distribution of these activation times, we'll have something like this. Then if we look at the DNA synthesis-related genes, they come a little bit earlier. But what about the positive feedback genes? If we try to locate where the positive feedback genes are, it's very interesting. All of them are in the earliest group. So remember this picture. So we have budding yeast and human cells which have different elements with different wirings, right? But are using the same logic. So in the decision point of the mammalian cell and the budding yeast, the proteins are coming from the different origins, and they're not orthologs, but they're functioning in the same way. So the principle is the same. They're activating the feedback first. Okay, so now we see that there's a high variation of temporal order, but what's the mechanism that regulates this temporal order? So the first thing to ask is, let's see what is the molecular basis of temporal order. So we remember that we have 2 transcription factors that regulate the the G1 to S transition, right? First one is the MBF, the other one is SBF. Then there are some genes that are regulated only by MBF. there are some genes regulated only by SPF, and there are some genes regulated by both. So if we look at the distributions of activation times within these specific groups, you'll see that, for example, MBF-only distributions, MBF-only targets activation distributions is like this. And if we put the SPF-only distribution, we'll see that SPF targets are significantly later than MBF targets in G1 block release. How about the mitotic block release? It's the opposite. The SPF is earlier than the MBF targets. All right, so it's switching. What happens to the dual targets? They are targeted by both SPF and MBF. If we look at those, we'll see that the dual targets follow the earlier activated transcription factors, right? So if you activate MBF, the dual targets are activated. If you activate SPF first, the dual targets still activate. So they're always earlier. That suggests the logical OR gate on the promoter. So that's simply, it is like this. When you activate one of the transcription factors, like in this case MBF, the dual targets turns on. And if you activate SPF, the dual targets turns on, regardless of the MBF being off. All right, so this idea comes from the batch experiment, right? We are looking at lots of genes, and we are inferring that there's a logical OR gate in the promoter. So we need to test it for the single gene. So to test it, we picked up a gene, which is gene 4, which is regulated by both MBF and SPF. So the case one is MBF here, and SPF is closer to the transcription start site. And what we did is we deleted either binding sites, so that in the wild type case, it's a dual target. If we delete MBF binding site, it becomes an SPF only target. If we delete SPF binding site, it becomes an MBF only target. And then we can test the same logic with the single gene level, on the single gene level, right? Then we monitored the GFP induction in the same way that I explained before, by aligning all the profiles to the budding time. Then remember that we had this from the batch experiment, including a lot of genes, right? We have, this is mitotic block release. This is the dual targets are following the SPF-only targets, and MBF-only targets come later on. And what we've seen for the single gene level is the same thing. That wild type means the dual target is following the SPF-only case, which is the MBF binding site is deleted. Then the MBF-only case comes later on. So if we look at the mean activation times, we'll still we'll see the same thing here in the bar plot, that the MBF-only case is delayed. Okay, so this was one case where we had the MBF, SPF, and the transcription start site. How about the other case, other way around? So now we have SPF, MBF, and the transcription start site in this XL2 promoter that drives the GFP. And we have done the same analysis. And we deleted both sites. And remember that this is from the batch culture. And when we look at the single gene level for this, we still see the same thing, right? The dual targets follow the SPF only case, where the MBF only case is delayed here, right? So the mean activation times are located here in the bar plot. So logical OR gate is still valid in this case. So remember that we have a switch case, like the order is switched in the G1 block release experiment from the microarray data, right? So dual targets are following the MBF only targets instead of SPF only targets in the G1 block release case. So to test this, to verify this is still valid in the single gene level, what we've done is we've arrested the cells by alpha factor in the G1, and then released from the G1 and then monitored with the GFP profile, GFP induction profile, and did the same analysis. And what we have seen is the same thing, which means that the order is switched. The wild type case, which is the dual target case, is following the MBF now instead of SPF case. All right. So, so far we saw that the budding yeast utilized the early induction of positive feedback, and combinatorial use of transcription factors helps to activate some genes earlier, such as CLIM1, right? in various environmental conditions. So whether, like, if the SPF is activated first, then Cleveland is turned on, or if the MBF is activated first, then Cleveland is turned on. Cleveland is positive feedback gene. Therefore, the cell wants to activate the positive feedback earlier in any condition. That's how they utilize the combinatorial use of the transcription factors. So to explain the molecular basis, we have seen that the combinatorial use of transcription factors explains some part of the variation. However, there might be some other factors that also affects the activation timing, the temporal order. So if we look at the single transcription factor targets, for example, here, look at the MBF, it still has a high variation, right, among itself. So there's still unexplained variation. So to target this question, we've tried some possibilities. For example, we've looked at the nucleosome positioning, if it has an effect on the transcriptional timing, and it turned out to be no, there's no significant effect. And we've also analyzed the promoters to see if the Tadabak-containing genes have earlier activation. No, we didn't see anything. Also, we've looked at mRNA degradation rates, which might regulate the activation timing. No. And also, we didn't see any effect of the multiple transcription factor binding sites. So we've eliminated all these factors. Then also remember that these transcription factors are already loaded on the promoter before start. So that means binding affinity is not the case here. So it suggests the question that maybe there's a global effect that regulates this the transcriptional variation, timing variation. So we know that the nucleus is not homogeneous. It's highly compartmentalized. And it's been shown that some parts of the nucleus is transcriptionally more active than the other parts. And some parts are transcriptionally silenced, right? So that might suggest that some part can be an early activated, like can have the early activated genes, and some parts of the nucleus can be late activated genes. So to approach this question, to answer this question, we might have two different approaches. The first approach, we can actually look at the similarly activated genes if they're colocalized in one place in the nucleus, in separate places. Or we can just look at one place in the different locations in the nucleus, and to see if they have different activation times. So luckily enough that we have a genome-wide chromatin interaction data that's developed by various labs. And based on this technique, it's called Hi-C for mammalian cells. But based on this technique, what you do is you cross-link the DNA, and you physically capture the physical interaction of the different parts of the DNA. Then you cut with the restriction enzymes, and you circularize it and further process to sequence it. And at the end, you have a piece of DNA where one side maps to one part of the genome, the other side maps to another part of the genome, so that you know that these two different parts are interacting in the physical 3D conformation. So at the end, you get a matrix like this, which records that each fragments are interacting with all other fragments with a certain value. But for our case, to get rid of the GC bias and length bias, we just converted every signal into the binary for stringent analysis. So what we have is, like, if two fragments have interaction, it's recorded as 1. If they don't, it's recorded as 0. So our idea is to look at the early activated genes and late activated genes, and similarly activated genes, if they are colocalized in the same place. So how can we do this? So we pick up a certain amount of gene, let's say 5 gene, right? Then we look at how many interactions we observed. Then let's say we observed 7 interactions. So what's the maximum possible interaction that can occur for 5? is 10, right? Then you divide it by 10, and then you have a score, which we call the colocalization score. Okay. So how do you know that this colocalization score is significant that says that these 5 genes are colocalized? We need to compare with the distribution, right? So what we did is we picked up a random 5 gene in the pool, and random 5 fragments, and look at their colocalization scores, right? And if we repeat this for a lot of times, we'll get a lot of different values, which give you a distribution. Then if you locate your the groups of interest in this distribution, the right-hand side area will give you the probability that it can be a random selection, right? Basically, that means the p-value. So based on this p-value, we can now classify the group of genes that we selected as colocalized or not. So what we've done is we've first ordered the genes based on their activation times in mitotic locally. Then we picked up n number of genes with a window size, let's say n, and then looked at if they're colocalized or not, with the p-value less than 5% or not. So if they're colocalized, we score them 1, and continue by sliding the window 1, and test it again, and score them 1 if they're colocalized or not, right? Then at the end, when you continue this with different window sizes, then you'll end up with this coverage plot. If you sum up all the scores for each gene, you'll come up with a coverage plot, which basically shows that the early activated genes and the late activated genes are separately colocalized, maybe the middle ones also. So that actually shows our idea that the early activated genes and late activated genes are colocalized separately. But this is, remember that this is coming from a mitotic localized experiment. If we do the same thing for G1 localized experiment, we'll see something like this, which says that the late activated genes are merely colocalized, maybe. But this is also valid because the high C data, the interaction data, is coming from an asynchronous culture, which is an unperturbed free-running cell cycle, right? Which we have already shown that the mitotic localized experiment is more relevant to the free-running cell cycle. So the G1 localized case is just a control in this case. Okay. So this was one way to approach that we've seen that the similarly activated genes are colocalized. The other way around is to look at some specific localized groups to see if their activation time distributions are distinct. So to do that, we need to cluster genes, right? Find out the clusters inside the nucleus. To find the clusters, we need a distant matrix. And how do you create a distant matrix with binary interactions? Then what we've done is we've assigned the 2 closest fragments to a gene for its like 5' and then the 3' end, and look at the binary, the pairwise interactions of these 4 fragments. So you can have 0, 1, 2, 3, or 4 maximum. So we have a distant matrix which ranges from 0 to 4, and which gives you the some proxy to the distance of these 2 genes, right? Then when you do this for our set of genes, you get some metric which is represented like this. And by using a hierarchical clustering algorithm, then cluster the genes, you get some a certain amount of clusters based on some threshold. Then if we analyze the activation times, compare the activation times of these clusters, we get the cluster 1 and cluster 2 have significantly separate activation times, meaning that the cluster 1 is very early than the cluster 2 significantly. So, but what is to do with this cluster 1 and cluster 2, right? Naming is not important here, so there must be something related to these cluster 1 and cluster 2. So to answer this question, first we tried how many amount of interaction that they have. What I mean by an amount of interaction is this: like one gene can interact with a lot of different parts in the genome, right? So total amount of interaction is means that total amount of different parts that interacts with. So we have seen that the cluster 1 has high amount of interaction, like it's interacting a lot through the, to the rest of the genome than the cluster 2, with significantly. So basically, the total amount of interaction separates these two clusters. Basically defines the property of these two clusters. Okay, then the question is, can we use this total amount of interaction to stratify the activation times? Yes. What we have seen is highly interacting genes have pretty much earlier activation time than the lower interacting genes. Actually, the total amount of interaction explains better than the clustering. So, okay, but what is the total amount of interaction, right? What's the relevant of biological relevance of this total amount of interaction? This is the obvious question that we can ask. Then what we know, I mean, think about this for a second. If you have a high total amount of interaction, that means this gene can interact with a lot of places, which means it's kind of like a flexible, right? It can, so the high-C data is coming from a sample culture, so you're getting a lot of different interaction bars. If you have a little amount of interaction, that means it's like stable in some point. So it can be related to the open or closed chromatin structures, right? Which we know that it's being regulated by some epigenetics markers like acetylation, methylation, and this kind of the histone markers. And it's already been known that if the chromatin is condensed and closed chromatin or euchromatin, the transcription, it's actually transcriptionally inactive. In order to activate the transcription, you need to open up this chromatin and make an open chromatin structure, which enables the more transcription. So basically, acetylation and methylation of these sites means that they have a repulsion of each other, and then they open up the chromatin. Actually, the acetylation, the methylation corresponds to the transcriptional activation, right? The open chromatin structure. So if we look at the genome-wide analysis of these the chromatin markers, first we look at this histone positioning, where we have seen that the histone poor regions have higher amount of interactions than the histone enriched regions. If we look at the acetylation marker, you'll see that the acetylation enriched regions are higher, have higher interaction than the acetylation poor region. All right. Remember that the acetylation is related to the transcriptional initiation. All right. How about the methylation? That's also true for the methylation, where you see that the enriched methylation for different sites are related to the higher amount of interactions. Okay. So they're just like asynchronous culture. So we're just looking at the total amount of interaction from the asynchronous culture, right? You mean the modification time scale? I don't think that. I mean, these are like somewhat global scale. Like I'm looking at the 5 kb, 10 kb regions. It's not like specific promoter regions. So these are probably stable. Like it's not so dynamic. So, if we put all the pieces together to make a big picture. So first, we saw that the cohort genes colocalize, and the colocalized genes activate similarly in both ways. And we also saw that the higher interacting genes have earlier activation times. Okay, so that means that open chromatin structure has higher interaction amount. And then, based on these three, we can say that specific locations have open chromatin structures. And does that mean that the specific location affects the activation timing? Actually, our data suggests that. But we need to confirm in a single cell level. So what I'm doing right now is trying to relocate the CLIM1 gene with its intact promoter to highly and low interacting loci to see whether the activation time is going to be changed or or not, right? So this is one experiment that's going on now to see if the activation time is being affected by the specific location in the nucleus. And second, I want to see the distribution of SPF and MBF in the nucleus with the super resolution imaging in collaboration with W.E. Werner there. So these are ongoing experiments that I'm pursuing now. And in summary, if we summarize all things in very limited items, I can say that, okay, we've seen that the cells first commit to the cell cycle, then transcribes the massive amount of, the transcription takes place. Second, we've seen that this is very important, so it's conserved in bioness and human cells. This principle is conserved. And in the second part, we've seen that the transcription acts like a logical OR gate, which explains the which partially explains the variation of the temporal order. And the fourth, we've seen that the nuclear sublocalization correlates with the transcriptional activation. So I would like to thank, first of all, to Jan, who supported me a lot during this PhD program in both ways, both academically and psychologically, or like, how would you put it? Like, he supported me a lot. Then I would like to thank the committee members, Steve, Daniel, Aaron, and Surya, for coming here and evaluating my research. And of course, I would like to thank all members of Skotheim lab who accompanied me throughout this journey. Then I would like to thank Andreas Gallman from Werner lab, who is helping me out to look at the super resolution imaging for the SPF and MBF PAM cherry strains. And also Amy Gleifelter and Corey for their collaboration in other project that Jan talked about. And Rob Debrin for his insights on my research. And also Lisa, thank you, Paula and Claire for helping me out some official documentation and this kind of stuff. And of course, the Turkish community helped me to engage more here. Here, of course, I didn't forget my family to thank here. So, of course, and also the funding agencies. So, thank you very much for listening to me. And yeah, I'll be more happy to answer your questions if you.
For the combinatorial circuits that you had, you said that you would take a look at whether having multiple numbers of the transcription factor binding sites makes a difference, and whether nucleosome favoring sequences makes a difference or not. Have you looked to see if there is any kind of a correlation between the transcription start site offset of the transcription site binding sites to the expression of the genes, both for the repressor and for the activator case?
Yeah, it's a great question. I looked at it. There's a slight correlation, but it's since it's explaining a little of the variation. It's not like too much. I didn't touch based on that. But yes, I mean, we have a limited amount of transcription start sites for budding yeast, not all transcription start sites. And some genes have multiple transcription start sites. So based on the data that we have a little correlation that if you have a closer binding site to the transcription start site, it activates better, but, I mean earlier. But, it's not so strong that we can claim that, okay, this is one of the major effectors.
Did you try engineering extra sites in and see if you could change the threshold, like change the size threshold to get even smaller or earlier?
You mean put extra MBF binding sites or something?
It is. Like if there's something else that's limiting.
I mean, since the since the multiple transcription factor binding site analysis didn't suggest us to do that, we didn't try. And if you would find something that, like, if you could, like, have 3 MDF binding sites which activate much more earlier, then we would definitely try, but it didn't suggest us.
I just have a question about is there anything known in Bionis about histone, the nucleosome positioning? Has that been done on a global scale there? And
Yeah.
can you use that also to support this in terms of evolutionary contribution?
Good question, but actually Bionis is not as well studied as Cerevisia, you know. So I don't know if they have. I don't look at the nucleosome positioning in the Bionis, but it'll be very cool to see if the, like, sequence, I mean, SNPs and these kind of stuff that affects the nucleosome position, which would relate to the activation. Yeah. It would be cool to look at. Yeah.
You didn't see a correlation with nucleosome position?
Oh yeah. I mean, in general. I mean, without looking at Bionis, no, we don't see it.
But
Yeah. All right. Thanks again.